I want to welcome you all to our 10th annual eye-opening conversation this evening that we started doing. We're going to hit around year 14 or 15 of the Byers Eye Institute, year 8 or 9 of the Mary and Sash Spencer Center for Vision Research. But I probably should have started by introducing myself. I'm Jeff Goldberg, professor and chair of ophthalmology. I'm a glaucoma specialist. So with that said, let me introduce the first of our speakers, Dr. Alana Grajewski, who we just recruited to Stanford last fall. And she is the absolute world leader in pediatric glaucoma. She comes to direct our pediatric glaucoma program. She brings to this actually a really important, impactful international flair of taking care of kids and adults with glaucoma throughout the lifespan, teaching people and helping build systems of care outside, even outside our community here. I want to thank, she actually had quite a number of guests who flew even internationally here to be with us today and to help us welcome her here to Stanford. And so it's a real pleasure. Alana, you're on first. Take us away.
Thank you for that introduction, and I am very proud to be here. I truly am. While I look at this group, I've been talking to a few people who are patients here as well, and I just want to get a feeling for those who I'm talking with at this moment. How many of you here in the room either know someone with glaucoma or have glaucoma or a question of glaucoma? Wow, okay. How many of you here know children and infants get glaucoma? Okay. Did you realize the percentage that is preventable for vision loss in both adults and children? Like 80% of visual loss in all age groups across the board is preventable and treatable. That's an amazing number. So one of the things that is a little tough, you saw very few hands raised for pediatric glaucoma, and yet it's the leading cause of blindness in the world in children. It's amazing. I mean, honestly, it is very different than adult glaucoma. In the incidence of pediatric glaucoma, I would say in the time that I was trained, and I was, pretty much always interested in glaucoma as a kind of chronic disease that was very similar to things I loved in internal medicine. But one of the things that just struck me was how everybody said this is uncommon and this is rare. And quite frankly, when you have a passion for something that is considered rare or uncommon, it's kind of a problem. So, I mean, I treat patients, as Dr. Goldberg said, with adult glaucoma. That's how I've been trained, as a glaucoma specialist in adults. But my passion has really been for trying to change how we all view children with glaucoma, at least in ophthalmology, in the medical field, and in the world. And yet, it was really underrepresented when I first started out. So much so that when I said I really wanted to build a clinic, they said, well, we're going to give you some space. And no joke, it really was a broom closet. And they really did believe that wasn't an insult. They really believed that's all I would need for how uncommon the disease was. But then again, this was my passion. It's hard to change that. So one of the first questions I'm often asked is, how do you even know if a child has glaucoma? So those of you who raised your hands about you have glaucoma or someone you know does, you have an idea that it has something to do with intraocular pressure, right? I see a lot of heads. And the thing is in children, elevated intraocular pressure manifests itself in a different way. And it has to do with the unique time period that it presents. anywhere from birth to 3 years of age. It is like a completely different set of symptoms. In adults, you don't see any change in the eye, do you? But everybody's looking at the optic nerve and the damage it might have. Well, in children, the eyes can either grow very large because of the elevated pressure. The cornea can become cloudy. The eyes are tearing, irritated. They don't like light. And quite frankly, at its latest stage, you can't miss it. You just can't miss it. So how do we treat it? Okay, so here's this, what was considered uncommon, and yet there was something about this that was very, very, very engaging to me. And that was that if we diagnose it early and we treat it appropriately, that child, if it is primary congenital glaucoma, stands a good 80 to 90% chance of having normal or near normal vision for the rest of their life. And if you don't, they're blind forever. So it's an all or nothing event, all or nothing. If that doesn't capture you as a physician who really has set their sights, so to speak, on preserving vision, I don't know what will. And it became very, very obvious that it was going to take a team to do this, that we were going to need to collaborate with people from all over the world in order to come up with those processes that would diagnose early and treat appropriately. So my team started off pretty small, and this is actually a big version of the team. Started off with about 5 people, physicians from all over the UK, the United States, some very, very notable people. And we started off by thinking, okay, we have an idea of what we think is the best things to do from our experience, and we're the ones doing a lot of these. Why don't we tell the world? And why don't we work with the rest of the world? So cutting to 8 years later, children's CGRN, the Childhood Glaucoma Research Network, was born, and we are now in every major eye center in the world and teaching the rest of the world. So does it matter? I mean, did we make any kind of impression on anyone? Well, the answer is, of course. I mean, you know, if it was your grandchild or your child, you'd relate to that one case. But the fact of the matter is, with burden of disease, with quality of life, with so many parts that vision plays in education, economics, socioeconomic mobility, it makes a huge difference, whether it's in the United States or whether it's anywhere. So global impact of that became a big part of what we were doing. And we started to say, we need to figure out, here in the US and everywhere else, how can we get these children? We can train the doctors how to treat, but how do we get the children in time to treat them? In the United States, vision screening is with the public school system. They're 7, 8. It's getting a bit late. And so using accessible technology and using an in-place healthcare system, and also with the support of some very, very generous people who have really the vision for children's future and what vision means to their education, have supported building this system. And I think we're making great progress. So now I'm here, and I want to build a center in Stanford. But why Stanford? It's obvious. I mean, honestly, the collaboration across disciplines, the whole name itself works to help us to create healthcare policies elsewhere. I don't really believe we should go in and only, do these clinics that come and go. No, we have to create healthcare policies where people recognize the importance of saving vision in children. So really, when you think about it, what I want to do is from that broom closet to the first pediatric glaucoma center that I built, I want to do more here. I want to create a center that is really towards visual impairment of all kinds in children, because early diagnosis makes the difference. So we're thinking bigger. Thanks to Dr. Goldberg. When I said what I wanted to do, he said, think bigger. I thought, wow, you betcha. Part of my life's in Minnesota. You betcha. And so that's what we did. And I've only been here a few months, and I will tell you, it's been definitely the right choice. These are our goals. Humbly, I state them because they look like they're, oh my gosh, a pie in the sky. But I can tell you, I've gotten that pie in the sky before. And the reward are the thankful children who you operate on at 2 weeks of age, who later draw you a picture with their eyesight with a condition that previously was routinely resulted in blindness. So I want to thank everybody here who supports this idea. Thank you for your time.
Thank you, Alana. Fantastic and really inspirational. Think about delivering just the top flight clinical care, research network, global impact that you bring. I next get to introduce Dr. Andreas Tolias. We're going to hear, he's professor of ophthalmology and also by courtesy electrical engineering. He really has brought together and married two amazing areas of research, which is to say, collecting data about how our brains process vision, how our cortex works to perceive vision, and then innovating on the computational tools, the AI. He's really the leader in a new field called neuro AI, which is really bringing these fields together to think about how do we see, how do we process vision, how can we leverage that knowledge to improve our perception of vision. This is just magnificent, impressive work. Up next, Andreas, thanks for joining us tonight.
Thank you, Jeff, for the nice introduction. Thanks, everyone, for coming here tonight, and thank you for the beautiful talk. So, about a third of our brain is actually specialized to receive information from our eyes and enable us to perceive the world. So although the eyes is the entrance to our pathway to see the world, you think about it, about a third of your brain right now is working without you trying or being aware of it, trying to make you see and perceive the world. It just shows how important vision is for us primates. And what I want to talk to you about today is a question and our path trying to see if we can heal the brain and enable blind people to see through their brain. And first, I want to ask, what is vision, or what is our brain really, what has it evolved to do? Our brain really, if you think about it, has evolved to build a model of the world and the universe outside. So what's happening inside our skull is a model of the world outside. And this is true about everything we do in our lives. For example, if we're trying to make a decision, what we want to study, what we want to eat, who we want to fall in love with, get married, see, it's all about trying to understand the outside world with our brain. And our brain is a very, very complex system. It encompasses about 100 billion neurons talking to each other, and you get about 100 trillion connections. So you can think of our brain as a society of neurons talking to each other. And try to understand the relationship between the outside world and what's happening inside our brain in vision. It's a very, very challenging problem. And let me explain you why. If you take this image, for example, this is a relatively low resolution images. On a computer monitor that is low resolution, you can generate more images than electrons in the entire universe. The entire universe is about 2 to the 300 power. So if you take 2 and you multiply 300 times, you get the number of electrons in the universe. On this screen here, you can generate on a computer 2 to the 65,000 possible patterns in the universe. So it's a huge number of images that the brain could, in theory, process to try and understand the world, and just tells you how remarkable and how complicated this is. So to try and understand it, our approach, as Jeff was saying, this new era called neuro AI or neuroscience and AI, we're trying to build a digital twin of the brain to then try and understand the brain using modern AI tools. So the idea here is that instead of just trying to understand it directly, we just collect a lot of data from the brain, then we build a digital twin of the brain, and that enables us, it gives us a new capability to scale up our experimentation and try to understand the language or the code of the brain. However, to do this, we have to sort of change our mindset, the way we've been doing science really, or neuroscience, for the last 100 years. And this is the lesson that we learned from AI. I'm sure you're all using these large language models, ChatGPT, Gemini, or others, or Claude. And one of the things that AI researchers figured out is that in order to train a model to build a model of something, in this case language, natural language, you have to train on the entire internet, right? The reason that these large language models work is because they are trained on the entire text that we humans in all languages have written in the last 4,000 years. But these kinds of, so basically instead of just training it on a subset of this text, these models are trained on the entire text. However, this kind of data that doesn't exist for the brain, because the way that, the system or our society has been organized, our funding, and the way we've been doing research for the last, 100 years, as I said, hasn't been to collect this kind of brain data. And here at Stanford, about 18 months ago, we launched this new project in the ophthalmology department and with collaborators from other departments called the Enigma project. And the goal of this project, the first goal, is to collect this data, is to collect the text, if you want, of the brain processing visual information. We're starting with vision. So if you look at this graph here, this is the only graph I will show. And here we see the brain coverage, how much of the brain we're recording from. And here is the complexity of the tasks that the brain does. If you think of vision, let's say it's all the types of natural images that exist in the world. Most of the academic research until now has been in this bottom left corner, right? And again, this is the way individual labs to very specific experiments to collect specific data to take specific hypotheses. So what we're doing now is through this Enigma project, we're in this trajectory to collect for the first time the brain level data set of text, the equivalent of text. And these are paired data between video, natural video, and neural activity at this very large scale. And to do this, we're partnering with the most advanced neurotech company in the world, IMEG in Belgium, that builds chips for the industry. And they have agreed to build these very fine wires that gives us about 2 to 3 orders of magnitude more large-scale data than even Neuralink, Elon Musk's brain machine interface company. So this enables us now to record this kind of data at a very large scale. And we build this entire pipeline that we're collecting the data, we're building these digital twins of the brain. And once we have these digital twins, now what would have taken us thousands of years to do experiments, we can run them in silico at very large scale. Because if you think about it, if I have a copy of a brain, then I can just parallelize it and have 1 million of these copies on a computer, on different computers, and around 1 million experiments simultaneously. And you can scale up your experimentation and then try and decode the language of the brain. Now, once we have this language, we're trying to follow two approaches to heal the brain, to get the brain, let's say, ideally of a person who is completely blind for different reasons, to write back down information into their brain, because the brain is still there. Even if you're missing your eyes or you have glaucoma or other diseases, degenerative diseases, the visual brain is still there and could receive information if we understand how to speak to it. So one is that we're building this chip in collaboration with Columbia University called the BISC chip that we can implant directly onto the cortex, and then using a camera system, we can write down the information into the brain by bypassing the eye in this case. The second one, and this is a new project that Jeff is starting with the Department of Psychology here at Stanford and Psychiatry, we're trying to open the plasticity window of the brain. What happens to us is that when we're young, our brain is very plastic, but then as we become adults or even, juveniles, then we lose the brain's ability to be plastic. And this is shown here. This is quite remarkable. But if you take an artificial neural network and you show it, you degrade this image and you bring it down to something that we humans cannot see now, it can reconstruct the original image. So the idea here is that if we open the plasticity window of the brain, even if it's receiving suboptimal information, it could regenerate, it could learn how to see again. Since the information mathematically is there in the image, even in a degraded image, because our perception at the end of the day is reconstructing what the world we think looks like. So I would like to acknowledge, the many collaborators and funding agencies that are funding these projects. And also I would like to thank you for coming here today. Thanks.
Thank you, Andreas. I can't emphasize enough how conceptually this is a new advance. At the Eye Institute, I do think that we are actually leading the world on how to treat the eye when you have eye diseases like macular degeneration or glaucoma, two of the really common ones in adults. But the idea that we could treat the brain to restore vision and say, hey, even if you're getting bad data coming from the eye or incomplete data from the eye, we could retrain your brain to make more use of that data, retrain the brain so that you can still perceive and see better again. It's just really remarkable. So Andreas, thank you for bringing this forward. Really, it's amazing. Coming back to the eye, I get to introduce Dr. Ted Leng. Dr. Leng is a professor of ophthalmology and a consummate retinal surgeon, but also a really big innovator, one of the real leaders in the field of stem cells. This would be a way to treat the retina in diseases like macular degeneration to restore vision to the blind. We have treatments that can slow down your loss, but to restore vision loss to the blind, Ted is really one of the first people ever in the country and the world to do stem cell trials for these diseases, and he's now leading not just in the clinic, but also in the laboratory. He's going to share that with us today in what is our very own TED Talk. All right, Ted.
All right. Thank you for the introduction, Jeff. So in my practice of retinal care here at Stanford, we have a lot of things that we can do to help our patients. But unfortunately, there are patients that continue to get worse despite the currently available therapies. One such patient of mine is a 78-year-old grandmother of 3, and I've been seeing her for over 7 years now for advanced dry macular degeneration. Every time I see her, she tells me that her vision's worse, and the disease is slowly encroaching into the center parts of her vision. She's worried that she will lose her independence over time, not be able to drive anymore to visit her friends for their weekly lunches or see her grandkids. Right now, there's nothing that we can do for these patients to restore their vision. There are many patients like mine. In fact, almost 2 million Americans have diseases like this, where they are losing or have lost their center's areas of vision, just like in the right photo here. They can't see what's in the center, and there's nothing that we can do right now to help them see better. Functional vision, like reading and driving, depends on cells in the back of the eye to be working and functioning all in concert together to derive those signals that Andreas is trying to decode. In diseases like macular degeneration, Stargardt disease, and retinitis pigmentosa, those cells are missing and dead, like you can see here on the right side of the picture. So our goal is relatively straightforward, but a lofty goal. It's to get a treatment for these patients who are suffering from vision loss as soon as possible, so that instead of missing the center of their vision, they can see again. We want to replace those missing and dead cells with neural stem cells. And let me explain what that means. These cells are human-derived, so they don't come from animals. They come from brain tissue. And let me remind everyone, in case you don't know, that the retina is actually an extension of the brain and the central nervous system. And stem cells, meaning that they can become any other type of cell in the central nervous system, including retinal cells. In atrophic diseases of the retina, the light-sensing cells are missing right here in the center. And we can image these in our clinics using specialized instruments like this picture you see here. The black areas in this photo are areas of those missing cells. And if you can't have those cells, almost like you're missing pixels in your digital camera sensor, the eye cannot see. Right now, there are 2 FDA-approved drugs that we use to treat advanced macular degeneration that are injected into the eye. You may have seen some of them advertised with the fawns here. But unfortunately, these drugs do not stop your vision from going away, and they actually don't restore the vision for these patients. They only slow down the gradual decline of the disease. So there must be something better for our patients. What we're trying to do is actually much more targeted than what's currently available. We want to put stem cells right into the area of need, right to this area where patients have atrophy and are missing cells. Our stem cells will go in there and replenish the areas where the native cells used to be before they died. For the first time ever, we've been able to design an innovative and effective surgery to reach all the way across to the back of the eye, under the retina, and inject these stem cells safely and effectively. But before we go into people, we need to prove that the cells work in an animal model. This is a rat model of retinal degeneration. And when the rats are born, they develop light-sensing cells we call photoreceptors, which are represented by these black dots in this red dash box. But these animals have a genetic defect. So left untreated or unaltered, they lose those light-sensing cells by 90 days. These little black dots are almost gone, and the animal cannot see. So the experiment we did is we put our stem cells in right here, these dark purple things. And at the same age of 90 days, what we saw was that the photoreceptor light-sensing cells did not go away, they survived. In addition to showing that the cells physically exist and survive in the presence of our stem cells, we also want to know what it did for the animal's ability to see. This is the same animal model of the rat, and this is a vision test that you do in the laboratory. Over time, left untreated, those rats again lose their vision. You can see this line going down very gradually over time. But when we put our stem cells into these animals, the rats retain their ability to see. So with this knowledge in hand, knowing that the stem cells both preserve the cells physically and anatomically in the eye, and they also preserve the rat's vision, we went to our first human trial of 15 subjects. The purpose of this trial was to determine the safety of the cells. And fortunately, we were able to show that the cells were very safe with no adverse effects at all the doses we tested. But the most exciting part was we also showed a signal towards improved vision in the eyes treated with the stem cells as well as a slowing of the rate of atrophy in those severe eyes. So the phase 1 study, by all measures, was a resounding success. But of course, there's always room for improvement. What can we work on? Well, the first thing was that the stem cells came in these clusters, as you can see in the photo here on the right. And these clusters required a larger needle than we would prefer to do the surgery. The clusters also prevented the cells from spreading out very evenly under the retina. Additionally, it took a lot of labor by hand in the laboratory to grow these cells, so the manufacturing process could be improved upon. So with generous philanthropic support and a $4.2 million grant from CIRM, which is California's stem cell agency, we went back to lab to make the cell product better. What did we do? The first thing we did is we converted the cluster formation into a single cell suspension. That will allow us to do a smaller surgery and also improve the spreading of these cells under the retina. We also reworked the manufacturing process, so now the cells are made in a more automated fashion using bioreactors like in this photo here. And this process is ultimately more scalable in the end. We took our new single cell suspension and repeated all those animal experiments I told you about and showed that both the cells were very safe and actually even more effective at preventing the deterioration or vision loss in the animal models. We had a great meeting with the FDA last year where we started to plan our next clinical trial. So, what do we want to support now? We want to support a new clinical trial at Stanford so that we can help restore vision for our patients. We are currently manufacturing the cells in our partner's laboratory in Southern California. We want to bring that manufacturing to the Stanford campus, to the laboratory for cell and gene medicine. We are going to do some additional cell stability and device compatibility testing, then complete and file a full investigational new drug application with the FDA so that we can begin our clinical trial at Stanford by the end of this year. So I'd like to thank you all for allowing me to share our exciting progress and future plans with you all. And especially I'd like to thank all of our current supporters here with us tonight. Thanks.